Hello, my name is Joshua Mugabi. I welcome you to Reproduction in Flowering Plants Self-Assessment Program. Which is the most accurate statement? The principal role of a flower in the life cycle of a plant is A. Attracting insects. B. Producing seeds. C. Producing pollen. D. Producing nectar. The most accurate statement is B. A flower's principal role is seed production. Statement A, C, and D are true of many plants, but these are functions which may help bring about seed production. Name the parts A to J shown in this drawing of a half flower of stitchworth. Part A is the stigma. Then part B, this is the style. Then we have here, this is the sepal. Part D, these are petals. Part E, these are anthers. And part F, it is a filament. Part G, which comprises of E and F, this is the stamen. And part H is the ovary. Part I, these are the ovules, and part J, it is the receptacle. What is A, the male gamete, and B, the female gamete in a flowering plant? A. The male gamete in a flowering plant is the pollen grain. Strictly, the gamete is the male nucleus in the pollen grain. B. The female gamete is the egg cell in the ovule. Complete the following paragraph by selecting words from the list below. Pollination is the transfer of dash from the dash to the dash in a flower. In cross-pollination, the dash from a flower on one dash is transferred to the dash of another dash of the same species. The list of words we are going to use: anthers, ovule, stigma, plant, flower, pollen, ovary, petal, style, receptacle, stamen. You can pause and fill in the blanks. Pollination is the transfer of pollen from the anthers or stamen to the stigma in a flower. In cross-pollination, the pollen from a flower on one plant is transferred to the stigma of another plant of the same species. Complete the following sentences. In a flowering plant, fertilization occurs when the dash of the dash fuses with the dash of the dash. After fertilization, the dash becomes the dash and the dash becomes the dash. In a flowering plant, fertilization occurs when the nucleus of the pollen grain fuses with the nucleus of the egg cell. After fertilization, the ovule becomes the seed and the ovary becomes the fruit. Which of the following statement is correct in flowering plants? A. Pollination can take place without fertilization. B. Fertilization can take place without pollination. C. Pollination and fertilization are the same. D. Pollination and fertilization must occur at the same time. Statement A is correct, though normally fertilization will follow pollination if the pollen and the stigma are compatible. Some species of plant are strongly adapted to pollination by certain insects. Which of the following characteristics would you regard as adaptation to pollination by bees? A. White or colored petals. B. Light, smooth pollen grains. C. Spiky or sticky pollen grains. D. Anthers and stigma inside the flower. I. Production of scent. E. Anthers and stigma protruding from the flower. F. Small green petals. G. Production of nectar. H. Production of pollen. Characteristics which are regarded as adaptations to pollination by bees are A. White or colored petals. B. C. Spike or sticky pollen grains. D. Anthers and stigma inside the flower. D. Production of nectar. I. Production of scent. Some of these features could equally well be adaptation to pollination by butterflies. A bee visits several flowers in succession on a single willow herb plant. In doing so, the bee transfers pollen from the young flowers near the top of the inflorescence group of flowers to the older flowers near the base of the inflorescence. Is this an example of self-pollination or cross-pollination? Transfer of pollen between flowers on the same plant is self-pollination. So is transfer of pollen within the same flower. Complete the drawing to show what has to happen before fertilization can occur. So here we have part of the flower, the female part of the flower. This might be the stigma of the ovule. We have the micropyle, and the ovary. The drawing should show pollen tube growing from the pollen grain to reach the micropyle of the ovule. So the pollen tube will grow through the stigma and it will enter through the micropyle going to the ovule. The drawing shows seeds of fruits of different plants. A. From the appearance of the structures, make a guess at how each one is dispersed, giving reason for your answer. B. What are the advantages to a plant of an effective method of seed dispersal? A, which is this one, it's a willow herb, and D, which is this one, Scalepias, have seeds dispersed by wind. They have got seeds which are dispersed by wind. The fine fluffy hair acts as parachute. The fine fluffy hair acts as parachute. Then we have E, agrinone, and F, herbibenet. These are dispersed by animals. The fruits have hooks which catch in the animal's fur. This is agrimon with hooks and herbibenet. They have hooks touch on the animal fur. B, this one, which is lime, and C, which is campion, are also wind dispersed. The lime has a bract which functions as a wing, slowing down the flutus rate of fall. This is the bract. It's going to act as a wing. The campion is an example of the scissor mechanism, in which seeds are shaken out of the fruit capsule when the wind sways the long stalk. So it shows a scissor mechanism. The root of pea seedlings is marked with equally spaced line as shown here. You can see the lines. That is the root of the seedling. Equally spaced lines as shown. Draw what you would expect to see in 2 days' time if a root A grew only from the tip. Grew only from the tip, this side. B, grew only at the top, this side. C, grew uniformly along its length. It was growing uniformly. And D, did not grow. So here it shows growth at the tip, meaning that this section here is elongating. And B, uniform growth, meaning that all the segments are growing uniformly. Then C, growth at the top. It is only the top segment which was growing. So you see it increased in length. and D, no growth. They remain the same as the original segments. What conditions do most seeds need in order to begin germination? B. What other conditions do the seedlings need to continue growth to mature plants? Part A. Condition most seeds need in order to begin germination. Most seeds need water, oxygen, and certain minimal temperature for warmth to start germinating. Then B, other conditions do seedlings need to continue growth to mature plant. To grow to maturity, the seedlings will also need mineral salts from the soil and sunlight for photosynthesis. How would you design, in principle, an experiment to test the hypothesis that a certain variety of lettuce seeds needed daylight in order to germinate? You would need a large sample of seeds, as was convenient for counting, say 30. This seed would be provided with water and suitable temperature. The seeds in the suitable container would be placed in a light-proof box or cupboard for a period long enough to allow germination. The box or the cardboard will not be opened during this period. The same number of seeds from the same bunch, same pocket, would be given identical conditions of moisture and temperature, but placed in daylight for the same period of time. At the end of this period, the number of seeds which germinated will, in each case, would be counted and compared. Figure 1 represents a pea seed split open to show its structure. Name the part A to C and state the function of each. Figure 2 represents a pea seedling 5 days after germination. Name the parts D to H. Figure 1, A is a plumule. This part here is a plumule and will form the plant's shoot. Then B is the radicle. This will form the plant's first root. Then C is a cotyledon. It stores food, mainly starch and proteins. Figure 2, D is a plumule. E is a tester. F, this is a lateral root. E is a tester. F, lateral root. G is a radicle, a root. H, these are root hairs. The early stage of germination takes place in the soil, where there is little or no light for photosynthesis. How does the seedling obtain materials for its growth and energy needs during this time? In the early stage of germination, the seedlings derive the material for its growth and energy from food stored in the cotyledon for dicotyledonous plants. You are asked to set up an experiment to investigate the effect of temperature on the rate of germination. You place 10 soaked peas in each of the 3 flower pots containing moist sand. 1 pot is placed in a refrigerator at 4 degrees Celsius. 1 is placed in a cupboard at room temperature, about 18 degrees Celsius. And the third is placed in an incubator at 25 degrees Celsius. You leave them for a week, checking each day that the sand is kept moist. How would you judge the results? B. Why was the pot at room temperature kept in a cupboard rather than on the laboratory bench? A. To compare the extent of germination at each temperature, you would need to measure the height of the shoots and the length of the roots of the seedlings from each pot. The measurement of each temperature would then be averaged and compared. You should also count the number of leaves and lateral roots, if any. B. Why was the pot at room temperature kept in the cupboard rather than on the laboratory bench? Because the seeds in the refrigerator and the incubator would be in darkness, it was necessary to keep the one at room temperature in darkness as well. Otherwise, any difference in germination might be attributed to the difference in light rather than the difference in temperature. Starch is one of the most common storage products in seeds. What happens to the starch before it can be used by the germinating seeds? Starch is insoluble and has to be converted by the enzyme to soluble sugar before it can be transported and used in the seedling. The diagram represents an experiment to test the hypothesis that seeds need oxygen in order to germinate. A. What is the liquid in A and what does it do? B. What is the liquid in B and what does it do? C. Which of the 2 flasks represents the control and what is its purpose? D. What results would you expect? From number 1, if oxygen is necessary for germination, and number 2, if oxygen is not necessary for germination. A. What is the liquid in A and what does it do? The liquid in flask A is a mixture of pyrogallic acid and sodium hydroxide in water. The solution of sodium pyrogallate. This solution absorbs oxygen and carbon dioxide from the air in the flask. B. What is the liquid in B and what does it do? The liquid in B is sodium hydroxide solution. It absorbs carbon dioxide from the air in the flask. Some experiments may use water rather than sodium hydroxide solution, but this is less satisfactory. C. Which of the two flasks represents the control and what is its purpose? Flask B represents the control. It shows that if oxygen is present, cress seeds can germinate even in these artificial conditions and also in the absence of carbon dioxide if sodium hydroxide is used. D. What results would you expect? Roman number 1, if oxygen is necessary for germination. And 2, if oxygen is not necessary for germination. If oxygen is necessary for germination, the cress seed should germinate in B, but not in A. If oxygen is unnecessary, the cress seed should germinate in both. What difference would you expect to see between pea seedlings grown for 10 days in total darkness and pea seedlings grown in the light for the same period of time? The seedlings grown in darkness will have long, thin, white stems, long internodes, and small unopened leaves. The seedlings grown in light will have shorter, thicker, greenish stem, short internodes. The leaves will have opened up and will be green and larger than those seedlings in darkness. Thank you very much for participating in Reproduction in Flowering Plants, a self-assessment project. Please do like, share, and subscribe.